I'm here to talk about a newborn screening study using a new DNA methylation sensitive high-resolution MELT assay on dry blood spots. Congenital hypotony represents a diagnostic challenge, especially in newborns, because it is present in several disorders, like metabolic disease, acute or chronic illness, genetic syndromes, endocrinopathies, and myopathies, and others. Besides, early diagnosis of Prader Willi syndromes provides improved treatment and accurate genetic counseling. The goal of our study is to establish the MS PCR technique associated with high-resolution MELT assay and assess the feasibility and accuracy of FPWR screening on dry blood samples. Here are the steps of our studies. The first step, we collected and stored blood on filter paper that we call DBS of 125 patients, 45 PWS patients, 1 Angelman syndrome patient, and 24 health individuals. The second step was the DNA extraction from DBS with 3 different methods. 3 commercial kits, QIAGEN, MAN, and Chelex. Those graphs are the comparison of DNA quality between DNA extraction methods. Initially, here in this first graph, the DNA extraction with MAN and Chelex provides a higher DNA concentration than the DNA extraction from QIAGEN. However, here in this 2 other graphs, when the DNA quality was assessed by different wavelengths, the QIAGEN strategy showed a higher DNA purity compared with the DNA obtained by MAN and Chelex, as you can see here and here. The step 3, so we can distinguish between methylation and unmethylation DNA. The bisulfide conversion transforms unmethylated cytosine in uracil, but methylated cytosine located with CPG islands remains as cytosine. The step 4 is the PCR amplification. We design a single pair of primers to evaluate the DNA methylation patterns of the promoter exon 1 region of the SNRP gene. It's important to say that only 1 pair of primers. It prevents the dimer formation, competition for the reagents, and it also serves as a control of bisulfide conversion, since it only works if the conversion also works. Here, to ensure the DNA quality of all clinical samples, the human gene RPP38 was tested. As you can see, all DNA samples was amplified. The amplification curves for RP38 for all DNA extraction here, as you can see, exclude the possibility of false negatives. The QIAGEN amplification curves display a lower CT, as you can see here in this first graph, than the MAN and Chelex, just a little bit. It's the QIAGEN is better. So the step 5, after the qPCR amplification, the HRM procedures starts increasing the temperature until the amplicons of the region completely dissociate of the double-strand DNA. We can detect different alleles depending on the CG-rich content. It is because of 3 hydrogen bonds between cytosine and guanine, and it needs a higher temperature to dissociate the double-strand DNA. As you can hear, the first is the unmethylated allele, and the methylated allele will require a higher temperature, as you can see here, than the unmethylated allele. So here are the methylation patterns for 3 individuals by HRM with unique pair of primers. Amplification plot related to health, this first line, the PWS and the AAS, the last one. The graph show the melting peaks here to each allele. Here we can see the unmethylated and methylated allele for healthy individuals. And for the PWS, we can see the absence of the paternal allele, and the Angelman syndrome, the absence of maternal allele. The Kaya gene works a little bit better. For all of them, the Kaya gene, man and child, it works, the amplification works, HRM analysis, but a little bit better for Kaya gene, because we can see these sharp curves without so many waves, as you can see here. We applied HMR technique to analyze the origin and proportion of alleles in a tumor cell line of the rat pituitary gland, GH3, whose genomic region 1q22 is orthologous to the human 15q1113 region associated with PWS. The GH3 strain was used as a study model for PWS using the CRISPR-Cas9 system, and it's a study in collaboration with Dr. Robert Nichols from Pittsburgh. So here we can see the karyotype analysis of the GH3 cell line confirm that it is a pseudo triploid cell line. The chromosome 1, which contains the critical region of PWS, presents in trisomy. We can see in the red arrow here the 3 chromosomes 1. So the evaluation by FISH of interface nuclei with 2 color probes visualizing the number of copies of chromosome region 1q22 in the cell line GH3. The 3 red signs here are the back probes inside the PWS region, and the 3 green ones are the back probe for control, just for control. This shows the trisomy state of the critical region of PWS. So the HRM enable the discrimination of parental origin, as well as the proportion of the paternal and maternal alleles of the genomic region 1q22 of the GH3 strain. So it was analyzed after bisulfite modification and show a double peak pattern due to a presence of unmethylated and methylated alleles of the PWS region. So the peak here, you can see the lowest dissociation temperature show almost twice the fluorescence intensity of the peak of the highest temperature, higher temperature. This result indicate 2 paternal alleles and 1 maternal allele in the GH3 strain in the critical region of PWS. The same thing that we saw in the karyotype and FISH, which indicates trisomy for the region. So we need to delete both paternal allele to silence the gene in the PWS region in the GH3 strain. So after genetic editing, the GH3 strain, we use again the HRM, so we can see these 2 strains. The 2 lines here had one of the paternal alleles, thus presenting disomy for the critical region of PWS, 1 paternal and 1 maternal. Despite the success achieved by the CRISPR-Cas9 methodology, another genetic editing event was necessary to delete the remaining paternal allele, and in fact, cancel the expression of PWS genes. So after the second round of genomic editing by the CRISPR-Cas9 system, it was defined 4 lines, here the lines 1, 2, 3, and 4, that show a deletion of the paternal allele, as you can see here, and null expression of the genes for PWS region. The other ones here, we don't have the maternal, it's just for the control. The advantage of the use of MS-HRM is listed here, so no further techniques required, for example, agarose gel, straightforward result interpretations. It's really easy to see the curves. And parallel samples analysis, we can analyze up to 96 samples at the same time. Low risk of cross-contamination. So the use of DBS sample as the main source of DNA provides several advantages against the use of the whole blood methodology. Demanding only a small amount of blood, ease of storage, ease of transportation, less invasive procedure, especially for hypotonic and newborns. That's not so easy to take blood from them. So in conclusion, our study demonstrates the potential of DBS as a DNA source for MS-HRM studies and its accuracy on abnormal and DNA methylation detection of imprinting-related disorders. I would like to thank my lab team in Fernandes Figueira Institution, the group from Endocrinology and Diabetes Institution, Dr. Robert Nichols from Pittsburgh, and the Brazilian Association of Prader Willi Syndrome. Thank you.